This video is going to cover some history of the discovery of cells. As you know, cells are really small, and so the discovery of cells is directly related to the use of microscopes, which is basically related to our ability to see things that are really small. We didn't know about cells really until we could see really small things, and we did that with the use of microscopes. Microscopes were first created by eyeglass makers, and that of course makes sense because these were people developing lenses. Some of the first microscopes in the 1500s, eyeglass makers realized that multiple glass lenses in combination can actually magnify really small objects to be easily seen. And they built the first true microscopes, one of which might have looked like this. And after that, nothing much happened until, in terms of what we'll normally talk about, until 1665, where Robert Hooke used an early microscope to look at a piece of cork. And so this is a drawing of what he saw. He saw all of these little tiny empty chambers, and he called them cells. So we will sort of recognize him as the person who coined the term cell. And today we know cells have lots of stuff in them, unlike what he saw in the cork of these sort of empty chambers. We're going to talk more about what cells have inside of them later. In the 1660s, Anton von Leeuwenhoek, so that's going to be really difficult, right? You have to think about Robert Hooke and Anton von Leeuwenhoek. Anton von Leeuwenhoek observed pond water under the microscope, and this is pond water that he actually normally drank. And he saw tiny living things in there. So he observed bacteria from a human mouth, and so then he sort of said he knew animal cells existed in animals and plants. And then between the 1600s and 1800s, not too many cell advancements that we're going to talk about were made. And the current view that was still widely accepted was this idea of spontaneous generation. If you break those words down, this is sort of out of nowhere and creation, okay? And so people believed that life arose spontaneously from non-living things. So for example, mice would come from dirty clothes, or maggots would come from rotting meat. And you have to understand that might seem silly to us, this sort of recipe for mice down here. That might seem really silly to us because we know that that's not true. However, at the time, people only knew what they could see. And what they saw was, oh, there was dirty clothes, oh, and eventually there's mice. Or if I leave this meat out, eventually there will be maggots. So therefore, the maggots came from the meat. So they knew what they saw, and that's what they saw. Ultimately, spontaneous generation was disproved by Louis Pasteur. This is the same guy who invented pasteurization, and pasteurization is involved in getting bacteria and other sort of microbes out of things like milk. And so spontaneous generation was disproved by this guy who was, again, looking at cells, looking at things that we can't see all of the time, looking at bacteria. In 1838, somebody named Matthias Schleiden, don't know how to pronounce his name, sorry, said that all plants were made of cells. In 1939, Theodor Schwann said all animals were made of cells. And in 1855, Rudolf Virchow said that all cells come from the division of pre-existing cells. And so all of these things together are summarized in what we now call the cell theory. And there are three main parts to the cell theory. The cell theory states, number one, all living things are made of cells. Okay? Number two, cells are the basic units of structure and function in living things. And now those two things might seem similar, but think about it. This is saying we're made of cells, and this is saying cells are the smallest bit of living material that you can have. So anything smaller than a cell, it would not be considered a living thing, basically. Does that make sense? Number three, new cells are produced from existing cells. And we'll talk about that when we get to cell division.